honored to receive this award. I'm so grateful to be here with all of you. So thank you. And I'm going to talk with you today about the work that I've been doing to better understand the genetic mechanisms of inflammatory disease. The rates of inflammatory diseases have been increasing all around the world for decades now, as we can see here with inflammatory bowel disease. So this really means that there's an already large and still growing number of people who need treatment. And there are treatments that are available. But the issue right now is that many of these treatments don't work for many patients. And so when we turn to the drug development pipeline as a new source of treatments, it's a really grim picture. Because only about 10% of the drugs that enter clinical development will ever become approved therapies. So put another way, over 90% of drug candidates fail. And when we look at why these drug candidates fail, over half of them fail because they aren't effective. And so it's important to clarify here to say the drug is effective in inhibiting its target kinase or other molecule. But it turns out that inhibiting that molecule doesn't stop disease processes and doesn't stop symptoms. And so this must mean that we're targeting the wrong molecules or the wrong pathways. Now, set against that very dark backdrop, we've had incredible success in disease genetics over the past 20 years. So with genome-wide association studies, or GWAS, we now have a really detailed understanding of exactly which places in the genome are directly involved in disease. And this has important implications for drug development, because it turns out that drugs that have targets that are genetically implicated in disease are far more likely to become approved therapies than drugs without this support. So it's anywhere between a 2- and 4-fold increase in the likelihood of success based on genetic evidence. So the question then becomes, if we know the genetic evidence is really important in identifying the right targets, why are we using it more? And the answer is that it's not straightforward to go from a genetic association to a better understanding of disease. And the reason for this is that the vast majority of genetic associations don't occur in coding regions of the genome, which is where they make predictable changes to protein structure or function. Most of them occur in non-coding regions, which can be really difficult to understand what they do, because even though we know they probably affect regulatory function, and the variants are actually usually associated with enhancers, we often need to figure out what the enhancers are doing. This isn't a straightforward problem, because enhancers often have a very cell type or cell state specific activity, and we don't always know which cell types and states are relevant to disease. And then on top of this, we also don't know which gene an enhancer is regulating. And so altogether, it's a really difficult problem. So I began studying one of these associations on chromosome 21. And the same set of variants is associated with multiple inflammatory diseases. So this includes ankylosing spondylitis, which affects the spine, both forms of inflammatory bowel disease, so Crohn's disease and ulcerative colitis, which both affect the gut, and then primary sclerosing cholangitis, which affects the liver. And when we look at the associated region, it doesn't have any genes in it. But we know that even though there's no genes, when we look at the diseases that it's associated with, they're all mediated by the immune system, despite affecting different tissues. And so the region was probably an enhancer, and then it was probably an enhancer in immune cells. So we went looking at H3K27 acetylation ChIP-seq data to look for active enhancers. We found one, and we realized that it was an enhancer that was active specifically in monocytes. Then we looked at promoter capture high-C data, which is this way of looking at physical interactions between bits of the genome. And we saw that it was interacting not with the nearest genes, so BRWD1 or PSMG1, but the much more distant gene called ETS2. And we also saw, based on expression data, that the monocytes of people who had 2 copies of the risk allele, there was higher expression of ETS2 than people without. So this is this nice correlative picture now, where we have a monocyte-specific enhancer, and it's driving expression of the gene ETS2. But we then need to know what ETS2 is doing. And so we started studying this by disrupting it. We would get monocytes from human blood and then introduce CRISPR machinery to knock out ETS2. We cultured the cells with inflammatory stimuli and then got macrophages. And from there, we started looking at macrophage function. And the first thing that we looked at was cytokine production. And we quickly saw that cells where ETS2 was knocked out were far less able to produce pro-inflammatory cytokines like interleukin 1, 6, and 8. Then we looked at phagocytosis, which is another key function of inflammatory macrophages, and again saw that without ETS2, cells were not able to pick up particulate matter very well. And we finally looked at reactive oxygen species production, which is one of the key ways that macrophages signal and function, but also a way where they directly drive tissue damage. And again, what we were seeing was that without ETS2, cells were far less able to produce ROS. So when we did RNA sequencing and looked at this transcriptionally, we saw all of the same deficits. So without ETS2, cells were losing activation, we're losing phagocytosis, losing pro-inflammatory cytokine production, and losing ROS production. And then interestingly, we also saw that certain metabolic pathways that we know are associated with a tolerogenic phenotype were upregulated. So all of this together meant that ETS2 was required for macrophages to be inflammatory. We then asked the corresponding question, which is whether ETS2 is sufficient. And this was a really important question to ask, because if you think back to the original association, people who have 2 copies of the risk allele have higher expression of ETS2. So we started studying this with a different system. We again got monocytes from blood, but this time made resting, non-inflammatory macrophages. And from there, we introduced mRNA that was encoding either ETS2 or its reverse complement as a control, and gave the cells just a low-dose inflammatory stimulus. And with this system, we get really nice dose-dependent overexpression of ETS2. And when we looked at RNA sequencing of these cells, the results were really striking, because we saw that all of the same pathways that were switched off when we knocked out ETS2 were switched on when we overexpressed ETS2. And they were all switched on in a dose-dependent way. So if we gave the cells a bit of ETS2, the pathway switched on. And if we gave them even more ETS2, we got even greater upregulation. And so all of this means that ETS2 is necessary and sufficient for inflammatory macrophage function, which very much positions it as a central regulator. So now that we have the central regulator of inflammatory macrophage function, it would be a really attractive therapeutic target. And the first step to showing that we can target it therapeutically is showing that it's actually present in diseased tissue. So we first did this by looking at single-cell RNA-seq data from inflammatory bowel disease. We looked specifically at myeloid cells from either healthy controls or from patients with disease. And what we could see was that there was a few clusters that emerged that were present in disease, but not present in health. It is most of those blue and green clusters. And the really striking thing is that if you keep your eye on those clusters, that's exactly where ETS2 and the ETS2 target genes are expressed. And then looking at the clusters to figure out exactly which cell types there are, it's clear these are both inflammatory macrophages and inflammatory monocytes. So now we can show that we have the highest levels of expression of ETS2 right in the cells where we'd expect to see it, and at the correct disease site. So we then went ahead and looked at ways to target ETS2. And the first thing that was clear was that it actually isn't possible to target ETS2 directly, because it's a transcription factor. It's not a druggable target that's kind of a classic druggable target. And there's no way to inhibit it allosterically based on its structure. So we looked for ways to get at this question indirectly. And the first thing we did was to go to the NIH links database. So this is the set of 30,000 cell signatures from cell lines that are treated with various small molecule drugs. And we looked for the signatures that were most enriched for the ETS2 signature. Then we grouped the different hits into classes. And at first, some of the classes that were coming out were approved IBD treatments. But the other key thing that we saw was that a lot of the drug classes that were emerging as hits were actually upstream regulators of the ETS family of transcription factors. And the largest class of these were MEK inhibitors. So we started testing whether we could use MEK inhibitors to modulate macrophage activation. And we first did this just with an in vitro setup. So we had inflammatory macrophages, we cultured them with a MEK inhibitor, and then did RNA sequencing. And this really nicely showed that we can downregulate key inflammatory pathways using this MEK inhibitor, including all of the same pathways that are downregulated when we knock out ETS2. And we then got one step closer to patients. So we got biopsies from patients who had active, untreated IBD, and then cultured them in the lab for about a day. And this is a really well-described system where the biopsies stay alive and they keep on secreting cytokines. And so that means that it's possible to test the effects of different interventions during this time. And in fact, when we looked at cytokine secretion from biopsies that were treated with MEK inhibitor versus a vehicle control, we saw there was downregulation of key pro-inflammatory cytokines really across the board. And the really interesting thing was these were a lot of key inflammatory cytokines that are targeted by current therapies. And we also looked at a transcriptional level. So we looked at the molecular inflammation score, which is a validated way of looking at disease activity, and saw that treating with the MEK inhibitor decreased disease activity, which again points us to this idea that if we can inhibit MEK and then inhibit ETS2, we can potentially help resolve inflammation. So what I've shown today is that it's possible to go from genetic association to a better understanding of disease mechanisms and to uncovering new therapeutic opportunities. This was really exciting work to be part of, so I want to thank my PI, James Lee, my other co-first authors, and the many people who helped collaborate on this work. Thank you.